I wanted to say something about Tylenol toxicity or acetaminophen toxicity. This is actually something that we looked at when I was doing research. After medical school, I did my internship and then I did a research year. And during that year, I was at a place called the House Ear Institute in Los Angeles. And we were basically, that's a place that helped develop the cochlear implant and technology, and a very well-known place of research. And I was working in a lab where we were studying the toxicity of different medications and trying to regrow the hair cells. So we were doing stem cell research. And they had a number of cases of patients who had been abusing hydrocodone or oxycodone, and we wanted to try to understand it. And so we looked at, usually they were taking a combination drug of hydrocodone and acetaminophen. And so we looked at that in our model. So what we did is we had cochleas from newborn mice, and we cultured the cochlea so you could see the actual spiral. And these were genetically altered so that you could see, so they fluoresced green under a fluorescent microscope. So you could see the hair cells, and then we would apply the toxic medication and see if there was an effect or not. And I got really good at killing hair cells, but not so good at regrowing them. It just became a toxic environment, so it was hard to regrow the cells. So anyway, we decided to look at this, and when we did it, what we found was this. Let me show you a couple of pictures. So when we applied the combination drug with acetaminophen and hydrocodone, it killed the hair cells. It was clear. When we separated out those two components, the acetaminophen and the hydrocodone, it was the acetaminophen that was the most toxic, and the hydrocodone didn't appear to be toxic alone. Now, we studied in a subsequent research study, we looked at hydromorphone, which is a metabolite of hydrocodone, and the hydromorphone appeared to also be ototoxic, but to make the Tylenol toxicity worse. Inside the cell, there's a structure called the endoplasmic reticulum. The DNA, which codes the genes for all of the proteins and all the things in the cell, is first transcribed, and another molecule called an mRNA is made, and that's taken outside of the nucleus, and then it's that message is used to create the proteins. Once the proteins are created, then they have to be folded into the correct configuration for their function. That folding takes place in this structure called the endoplasmic reticulum. One of my partners did subsequent studies after our research, and they looked at the mechanisms of the ototoxicity of the acetaminophen. And what they found was the acetaminophen and one of its metabolites, NAPQI, were both toxic by two different mechanisms. One, it increased reactive oxygen species or oxidative stress on the cell. And number two, it put stress on the endoplasmic reticulum, or the protein folding mechanism of the cell. And by both of those mechanisms caused toxicity to the cells. We had the same question that people are having right now when they're talking about Tylenol toxicity and the possible connection with autism. Why have we not seen this before? Everybody knows that Tylenol is toxic to the liver. When taken at high doses, people can overdose and die from acute liver toxicity. So why were we seeing this in the patients? We did a review of all the patients, and many of those patients had been abusing the hydrocodone, taking Lortab or Percocet, and abusing at high levels, and taking as many as 30, 40, 50 pills a day. And it had the Tylenol in that. So why were they not having liver issues? Why was hearing loss the only symptom? We believe it was because they gradually increased that dose over time. Their liver adjusted, increased its enzymes, was able to compensate to avoid the liver toxicity. But eventually the inner ear, which is part of the central nervous system, was affected. We also looked at many patients who were taking standard doses. So when you're prescribed after surgery, Lortab or hydrocodone or Norco, whichever brand it is, with the hydrocodone and the Tylenol, basically you can take 1 to 2 pills every 4 to 6 hours. And the reason why we dose it that way, we want to make sure that they don't get too much Tylenol, because you can get to the max of Tylenol if you're not careful. So many of these patients who develop the deafness, then they were taking the standard dose. They weren't taking more than the max dose per day, but they had been taking it for chronically for months. So one conclusion is that it's possible that Tylenol could be toxic to the fetus if the woman taking it is taking it either at very high doses or at doses that are standard doses, but taking it a lot over time. And that's very possible. We also know that the central nervous system is developing during the full pregnancy. The inner ear, for example, it starts to develop around week 4, but it continues to develop during the first, second, third trimester. But we know with our experience with other medications that are toxic, potentially toxic to the fetus, that that the cells are more susceptible, or the fetus is more susceptible during the first trimester. So if there's anything that has any potential harm, that's where the harm is the greatest. So one very easy deduction we could make, or one care we could take, is to not take any medicine that has any potential of side effects during the first and maybe even the second trimester. So anyway, the inner ear is an extension of the central nervous system. The Tylenol has been shown to be ototoxic, in other words, neurotoxic, when taken at standard doses chronically or at very high doses. And in our cochlear newborn cultures was shown to be toxic to the inner ear and cause hearing loss. So after we found that Tylenol was toxic in our cultures, we also tried to do this in live animals, and it's called in vivo model. So we had mice and we were exposing them to increasing levels of the Tylenol. And we were supposed to do this for a certain number of weeks and then test their hearing. Well, all the mice died. So we think they had liver toxicity and they died from the Tylenol exposure. So we weren't able to publish that. We also tried to do it in guinea pigs, because guinea pigs are, they have, the guinea pigs are used commonly for studying the hearing. We can test their hearing. And we put the Tylenol and the hydrocodone in the water for the guinea pigs, but we never saw any changes with the hearing in this model. And we think that actually the guinea pigs were like avoiding drinking the water when it had the Tylenol in it. And so they were, I guess, self-selecting to not have the toxicity. So we weren't able to reproduce it in that. My conclusion is, if in a developing fetus during the first trimester, even the second and third trimester, we should always take caution with any medications. And Tylenol is known to be neurotoxic, and so therefore we should take precautions. And maybe we can do some really good studies now. Well, you can't do a study with exposure, but yeah, this is so hard to study, because once you know that there's a problem, you don't want to expose people to the medication in order to study it. Otherwise, it's unethical.